Well, welcome everyone to this national chapter webinar on clinical trials. We're delighted you could join us this morning, afternoon, or evening, wherever you are. We have a great lineup, some great panelists, and other guests. I was just watching the chat as people were getting situated and logging in, and we have truly an international audience. I just wrote down some of the places that people are tuning in from: Slovakia, Ireland, Malaysia, Peoria, Newfoundland, India, Switzerland, Canada, and even Hawaii. So very exciting for us. To kick things off, I'll be back with you in a few minutes, but I'd like to introduce the Denver chapter president of the Foundation Fighting Blindness, Ms. Lindsay Blankenship.
Hello, and welcome to the Foundation Fighting Blindness National webinar on clinical trials. I am so excited you all were able to join us today, and we have an outstanding group of experts on our panel as well. Like Ben said, I serve as the Denver, Colorado chapter president. The Denver chapter was actually founded in 1978 by Bob and Beverly Martin. Since then, the chapter has done quite a lot of amazing things. Our vision walks are very well known and well populated, and since its inception in 2006, we have raised over $1.8 million. The other event that our chapter is well known for is the annual Scramble for Sight golf tournament. This year will mark the tournament's 21st year and has raised close to $2 million. So we're very proud of these events. I personally was introduced to the Denver chapter when I was about 10 years old at a support group meeting that Bob and Beverly held out of their home. And about 5 years ago, rejoined as an adult, where I brought back the support and networking group that now has over 100 members. And I have served as the president for almost 3 years now. I would like to thank my fellow leaders in the chapter, Megan Winthrow, Andrew Hurst, and Larry Young. They are fabulous individuals and wonderful leaders, and I'm very grateful for their friendships and all that they do for us. I am also very proud to be a part of this organization that is truly changing lives. I am excited to be a part of this wonderful session today, and it's now my pleasure to introduce Ben Shaberman, Senior Director of Scientific Outreach and Community Engagement of the Foundation Fighting Blindness, where he will give an overview of today's webinar. For 16 years, Ben has been reporting on retinal research for the Foundation's electronic and print publication. In addition, he presents the latest scientific advancements at local chapters and national events for parents and families, and conducts various training activities for staff and constituents. He enjoys working with constituents one-on-one to help them understand the retinal disease and the research underway that may benefit them. Welcome, Ben.
Well, thanks, Lindsay. I appreciate the formal introduction. It's great to be back again. First of all, I want to thank the thousands of people, both in the US and our chapters and various communities, and then people all over the globe who support our mission in a variety of ways, whether it's through donations or volunteer activities or other leadership activities. All the research that you read about on our website or hear through educational events like this is made possible by people like you. So thank you very much for the hard work and passion that you bring to our mission. Just to give you a quick overview of our program today, I will, in a few minutes, be providing an introduction to clinical trials, more about the process, the definition of clinical trials, the goals, and things like that. And then we'll have presentations from our three panelists. And after they're finished, we'll have somewhere in the neighborhood of 30 minutes for questions and answers, which you can enter into preferably the Q&A or the chat, and I will moderate that session. So to kick things off, I am delighted to introduce our the Foundation's new Chief Scientific Officer, Claire Gelfman. Now, I was just thinking, how long has Claire been with the Foundation? Because it seems like a very long time because of all the work you've done, the enthusiasm and insights you brought to your role, but you haven't even been here a month yet. So I'm very impressed. Let me give you a little background on Dr. Gelfman. Before joining us less than a month ago, she served as Vice President of Pharmaceutical Development at Adverum Biotechnologies, where she led preclinical efforts for the development of gene therapy-based treatments for retinal disease. She received a PhD in biochemistry from the University of Texas at Austin. Her foray into ophthalmology began during her postdoctoral fellowship at the University of California at Davis. Since then, she's held various research and development positions spanning both academia and industry at the cross-section of ophthalmology and molecular genetics. Dr. Gelfman brings more than 20 years of experience in ophthalmic drug discovery and development, including gene therapy approaches in both biotechnology companies and contract research organizations. So welcome to your first official event, Dr. Gelfman.
Thank you very much, Ben. It is a true pleasure and an honor to be here today representing Foundation Fighting Blindness. It's also a very exciting time to be in the field of ophthalmology. And I think what really excites me the most are the variety of approaches that are being taken right now to develop therapies that have the potential of really impacting affected individuals with inherited retinal disease. And I say that really spanning all sorts of types of therapies, whether that be a small molecule drug, a protein biologic, or even regenerative medicine. By that, I mean cell therapies and gene therapies. And when we think about gene therapies, we think about giving a quote unquote good copy of the gene back to restore function. And that's gotten a lot of traction. We're going to hear a lot about that today. I'm also excited by what are called gene agnostic approaches, where you can use a therapeutic to treat a rare disease, but it can treat many mutations, not just a single one, because we're getting at the mechanism, the pathology, not just bringing back a good copy of the otherwise malfunctioning gene. I'm also excited to learn about gene editing, these molecular scissors we hear about that are being used to go in and fix at the molecular level what is going awry as a treatment modality that's also being pursued. And then, of course, in the area of cell therapies, we hear a lot about researchers and companies that are literally giving back the missing cells to recreate the environment for a functioning retina. And sometimes it's giving back the cells, sometimes it's giving back cells that can make factors that are needed for the existing cells to work properly. So there's all sorts of different ways that are being approached. And I'm encouraged by this multi-pronged approach. And whether or not, no matter the approach, there is a continuum, there is a spectrum of drug development that starts at its inception of this idea. What is the gene that needs to be altered, that needs to be fixed? How do we go about studying that? And once we learn about that, we can take that information and begin to think about what type of therapy would work best. And then we start working on animal models. We start working on safety. We look at a model of the human condition, and we try to mimic that to learn more about treatment. And then once we get further down that pathway, if you will, then we begin asking questions that the FDA, that the regulatory agencies are going to want to know from us, so we can make sure that what is progressing as a potential therapy is safe, and that it's efficacious, and that it works in the patient population that it's intended to treat. And so that whole continuum of developing therapeutics for the treatment of inherited retinal diseases, the quote unquote last step prior to approval are human clinical trials. We've learned all this information about the biology, about how to deliver the therapy, about how much of the therapy to deliver, how high can we go and still for it to be safe, but working in the best way possible. All of that information is collated. It's given to the regulatory agencies in the United States, to the FDA, in Europe, to the EMA, depending upon where in the world you're seeking approval. And the hope is that there's been enough information that is gleaned, and all the work that the foundation supports is all about getting enough information to de-risk the programs towards approval, to poise them for success. And so the human clinical trials that are asking two important questions: is it safe, and does it work? That's what human clinical trials is all about. And so I think with that introduction, Ben, I will pass the baton back to you for you to give us an introduction to human clinical trials.
So thank you, Claire. That was a nice introduction to my introduction. And let me share my screen. And I'll be spending about the next 15 minutes just talking about some of the clinical trial basics. My last slide, I will review some projects that are either in the clinic or moving toward the clinic. But as I said at the beginning, I think the big takeaways from this entire webinar are more about the clinical trial process and what patients and families can expect participating in a clinical trial. Before I officially begin here, I would like to thank the many sponsors that make our webinars and many educational activities and other events possible. Our visionary champion partners include Genentech and Janssen. Our outreach gold partner is Applied Genetics Technology Corporation, or AGTC. Our outreach silver partners are Apellis, Astellas, Biogen, Mira GTX, and Spark Therapeutics. And our outreach bronze partner is Johnson & Johnson Vision. So again, thanks to all our generous sponsors. So let's start off with a basic definition of what a clinical trial is. And simply put, a trial is research in humans. And in many cases, this is for an emerging therapy, a treatment, a cure, a prevention. And on my slide, I have the word emerging underlined because that's an important qualifier. When a drug or a stem cell therapy or whatever is in a trial, it is not a therapy yet. It only becomes a therapy once it gets regulatory approval from the FDA or a regulatory agency like the European Medicines Agency. So we always say that when these approaches are in the clinic, we call them emerging therapies or potential therapies. Those are important qualifiers because we're still learning about them. And because of that, for those of you that participate in clinical trials or thinking about participating in clinical trials, you're truly pioneers because you're helping the researchers and the trial sponsors better understand if these treatments really work. When you participate in a trial, you may have the benefit of getting some vision saved or restored, but perhaps not, because we're still learning. A lot of trials, some people get placebos. So in that case, obviously, you probably won't have vision improvement. So I think this is one of the most important takeaways from our webinar today, is that human studies are research. We're still learning about the treatments. And as Dr. Gelfman said, safety is a really important goal in evaluating a treatment. The FDA is all about protecting the consumer first, and it happens at every stage of a clinical trial. then obviously you hope that whatever treatment that is being evaluated works. And we develop what we call endpoints or outcome measures to see if the treatment is meeting the desired goal. And in our world of retinal diseases, where they affect the retina and vision in myriad ways, developing these endpoints can be somewhat challenging. And maybe those would be good topics to dive into further with our panelists. Trials are very expensive, exponentially more expensive than the lab studies that make them possible. Perhaps a small trial for a rare disease, we could get away with spending tens of millions of dollars, but often these trials cost hundreds of millions of dollars. A trial is typically somewhere from 7 to 8 years, more or less, in duration. They take a while. And for you, the patients, they require a strong commitment. If the trial is out of town, you're going to have to travel on a regular basis. The good news is most of the trial sponsors will pay for that travel. But regardless of where the trial is, the patient is going to need to make many visits on a regular basis. And the visits themselves, the exams and all the tests they run, can be pretty grueling. It's not just a quick 15 minutes in and out. A visit can last for many hours. And then finally, just a little sobering comment is, by nature, clinical trials don't always lead to the outcomes we're looking for. In fact, most therapies don't cross the finish line. This is just the reality of clinical trials. And we have been reporting that we have more than 40 clinical trials underway in our space, which is wonderful. But not everything in those trials is going to cross the finish line. And that makes it incumbent upon us as a community to get dozens and dozens of more therapies into the pipeline, because not everything is going to work. So just to quickly review the different phases of a trial, trials start off really looking at safety. Safety studies, these phase 1 studies, are small, they're relatively short. And again, safety is the goal. If safety is met, which in many cases it is, then we move into a phase 2 and we start looking at efficacy. But also in a phase 2, we're exploring dosing, finding the optimal dose. Usually this is through dose escalation to find out what the highest and most effective tolerable dose is. And you'll have more patients in a phase 2 trial than the phase 1. And if we're getting some good signals in a phase 2, then hopefully the sponsor will move into a phase 3. We call this the pivotal phase, because if the treatment performs well in a phase 3, then it's likely the sponsor will seek regulatory approval. So again, in a phase 3, we're looking at safety. We're definitely interested in a strong signal for efficacy. And there are more patients enrolled in the phase 3 than in the phase 2. And these phase 3 trials are typically multi-center. So they can be at multiple sites around the US or perhaps even internationally. And the hope is that the different centers can replicate results, which just provides further evidence that the treatment is working. And then if things have gone well in the phase 3, as I said, the sponsor will look for hopefully regulatory approval. Now, as you hear about different trials, you'll often hear that phases are combined, and that's done to reduce the patient enrollment requirement or even speed up the duration of the trial. So I wanted to take a moment to talk about translational research, because this is an important topic. Translational research is simply the definition or the name we give for moving research out of labs, out of cell studies or animal studies, into humans, into clinical trials. And industry uses the sort of dubious phrase for translational research of crossing the valley of death, because a lot of promising therapies don't make it out of the lab into clinical trials because crossing that chasm is challenging. The bar for conducting human research is simply much higher than that for conducting animal research. There's a lot more risk, a lot more cost. Simply the standards for quality and safety are much higher in a human study. And once you move into humans, you have to work with the regulatory agency like the FDA, who is monitoring the trial the whole way. You need authorization to start the trial. The FDA is monitoring the trial the whole way. And then you need approval from the FDA if your therapy is working so you can bring it to market. And manufacturing of the potential treatment is really important. You need to employ what we call good manufacturing practices to ensure that the treatment is consistent and safe, and that is not a trivial thing. So the Foundation Fighting Blindness actually does a number of things, funds a number of research studies and approaches to try to help researchers cross this quote unquote valley of death. One of our programs we call the Translational Research Acceleration Program, or TRAP. This is a program where we help researchers get their emerging therapy to the clinical trial doorstep. And actually, Dr. Young, who you'll hear from, has really taken advantage of that program. Another program that we have that we're very excited about is our Venture Philanthropy Fund, which we call the RD Fund. And this is a partnership between us and venture capitalists and startup companies in most cases to get treatments that are near or in early stage clinical trials to the point where we can attract outside investments from big pharma and biotechs. And so to finish off here, I wanted to talk about some of the projects in our RD fund. And I'll note that our RD fund was started in 2018. I don't think it's quite 3 years old yet. And we have more than $70 million in assets for about 10 different projects. So let me go over a quick review of some of those projects. Cena Therapeutics is a small startup company that started out of the University of Florida. Researchers Shannon Boy and her husband Sanford are the co-founders of Cena. They have two projects underway right now, a phase 1/2 clinical trial for an LCA1 GUCY2D gene therapy. LCA causes severe vision loss in children. In case you didn't know that, this gene, GUCY2D, is one of the more common genes that can, when mutated, can cause LCA. And excitingly, even in this early stage trial, they've had some encouraging results. The first few patients at a low dose have shown some signs of efficacy. So Shannon Boy is also developing a gene therapy for people with USH1B that's caused by mutations in MYO7A. What's really cool about this approach is it's a dual vector gene therapy. About a third of the genes that we're trying to address are big, and they're too big for the gene therapy delivery systems that we're currently using. So Dr. Boy is coming up with a dual vector system to deliver these genes in 2 containers for, again, these conditions where large genes are the cause. So that's still at a preclinical stage, but if this works, it holds potential to help people who have mutations in other large genes. Another partner of ours is Nacuity. They're out of Dallas. They're developing a small molecule, an antioxidant for retinal diseases like Usher syndrome and RP. It's a very powerful antioxidant. If you can mitigate oxidative stress in people with these conditions, you have a good chance of saving and preserving vision, or at least slowing the disease down. It's in a phase 2 trial in Australia right now, and hopefully they will move this into the US before too long. What's nice about this is it's gene agnostic. It's not targeting a particular gene, so it holds potential for helping a broad range of people affected by these conditions. Another gene agnostic approach is being developed out of Cena Therapeutics, a startup founded by Tom Ray at the University of Washington in Seattle. Now, this is a really innovative approach. It's a small molecule that's designed to make rods look less rod-like. It changes their identity. So let me tell you why that's important. In a lot of retinal diseases like RP and Usher syndrome and others, it's rods where the gene mutation is causing the degeneration. And after a lot of rods are lost, the cones start degenerating. And cones are actually the most important photoreceptors for our activities of daily living. They give us the ability to read and recognize faces and see in lighted conditions, perceive colors. So what Dr. Ray is trying to do by changing the identity of rods to be less rod-like, he's preserving those cells, which in turn preserves cones. So it's a very innovative way to potentially preserve cone vision. This too is gene agnostic, and it's still at a preclinical stage, not quite ready for prime time, so stay tuned for that. Another really promising project that we're funding is for the company ProCure out of the Netherlands. So sometimes researchers try to address mutations in our genetic messaging system. So we have our DNA, which is like our genetic library, and then our genetic messaging system, which our cells read to make the proteins that our cells need. So by addressing RNA, we can potentially get the proper protein expression. I know that's a simple explanation for a complicated topic, but what ProCure is doing is they're developing this tiny piece of genetic material to cover up the mutation that's being carried along in RNA. They've shown early efficacy in their trial for people with mutations in the region exon 13 in USH2A. It's actually shown efficacy in people with later stage disease and earlier stage disease. So thanks to the good results in that phase 1/2, they're planning a phase 2/3. I'll just note, this is the project we're funding. ProCure also has projects underway for a gene called CEP290. That's moved into a phase 2/3 because that's worked well. They've had some really nice efficacy results there. And they also have a project for the gene rhodopsin, which has not reported results yet. So moving down the slide a little further here, I'd like to tell you about our partner Sparing Vision out of France. They have two approaches that are targeting cones, and I've already mentioned why cones are so important. Both of these projects are gene agnostic. One is designed to preserve cones for people with conditions like Usher and RP. That's still at a preclinical stage, but it's pretty far along. We're hoping they can get that into a clinical trial before too long. Again, gene agnostic. Now, another approach for therapy they just acquired from a small company goes beyond gene preservation. It's designed to add function back to cones that have stopped working but haven't fully degenerated. I'm calling this cone resurrection. So this is at even an earlier stage of development, so stay tuned for that. And perhaps someday, if both of these approaches work, they can be combined so that we can both preserve cones and bring other cones back to life. Another partner in our RD fund is Stargazer. They're developing a small molecule to slow the progression of Stargardt disease by slowing the uptake of vitamin A byproducts that ultimately lead to the damage in Stargardt disease. That's in a phase 2. Stay tuned. Hopefully we'll hear results from them before too long. And then finally, I wanted to mention the company Videre, who is developing an optogenetics therapy. In short, optogenetics is an approach designed to bestow light sensitivity to the retina after it's lost all its photoreceptors. Thanks to the attractiveness of Videre's approach, they were acquired by Novartis a few months ago. But there has been another Videre, we call it Videre 2, that has been established, and they're part of our portfolio as well. And they have yet another optogenetics approach that they're pursuing. So stay tuned on Videre 1 and Videre 2. So to officially conclude my talk, I want to remind everybody, if you want to learn about these therapies or other therapies, there's articles on all this stuff at fightingblindness.org. You can learn about our genetic testing program, the diseases, other educational resources at our website. Clinicaltrials.gov is a great resource for learning about the clinical trials that are underway. And we strongly encourage you to register in our MyRetina Tracker registry at myretinatracker.org to get on the radar screen of companies and researchers that are launching clinical trials, or at least recruiting for clinical trials. Again, register at myretinatracker.org. So let's take a breath, take a little stretch as we move into our next presenter. And I'm going to start, or I should say, stop sharing my screen and invite Paul Yang to start. Paul, you can take control of the screen, but let me give you an overview, a bio from, oh, I'm sorry, it's not Dr. Yang, it's Dr. Anglade who goes next. I'm sorry, I messed up the order there. Sorry about that. So I will give you an introduction to Dr. Anglade. He is an MD, Vice President and Clinical Lead of Retina at Janssen R&D US. He's responsible for clinical development of Janssen Pharmaceuticals' retinal disease assets through all stages of clinical development, including therapeutic strategy and protocol development. He's a core member of the Retinal Disease Area Stronghold, and working in close partnership with other leaders, he develops the clinical therapeutic area strategy for Janssen's retinal disease programs to ensure the timely completion of key deliverables to health agencies. Eddy is a senior level pharmaceutical executive with more than 20 years of experience designing and implementing clinical development programs and regulatory strategies. So, Dr. Anglade, it's all yours.
Thanks very much for your kind introduction, and I'm really delighted to be here today. I'll share my screen and just start the presentation momentarily. Hopefully you'll be able to see it. Let me know, Ben, if we're good. You should be seeing it shortly.
I see it well. Thank you.
Okay, great. Thank you. Full screen version?
Yes, sir.
Okay, terrific. So Ben, thanks very much, and I was really delighted to know of Dr. Gelfman's joining as well. Good morning and good afternoon. Thank you for your kind introduction. I'm Eddy Anglade, Vice President and Clinical Lead for Retina at Janssen, the pharmaceutical division of Johnson & Johnson. I'd like to thank the Foundation Fighting Blindness meeting organizers for the opportunity to meet with you and esteemed colleagues today, and also a thank you as well to those folks who are joining from many time zones away in the middle of the night. I'm excited to be here today to be able to provide you an overview with Janssen's portfolio and delighted to introduce you to our company and provide some details on our commitment to advancing much needed treatments for eye conditions. At Janssen, we pride ourselves on healthcare compliance, and because all of our research and clinical trials are in development currently, I will not be able to discuss them today or take part in the Q&A. That said, however, please know that your voice, the patient and caregiver voice, is at the heart of all that we do. As the largest healthcare company in the world, J&J Janssen has a legacy of innovation across many therapeutics areas and disease states. We are actively exploring ways to bring potentially sight-saving treatment to people living with retinal diseases. We aim to make a difference by discovering and developing transformational therapies that have the potential to advance the care of sight impacting conditions. Our commitment in this space is driven by the enormous unmet need for patients, society, and healthcare system. That's why we've taken on the challenge of developing new therapies for retinal diseases, and we're rapidly amassing new capabilities, new assets, and new partnerships. Our legacy in the ophthalmic space puts us in a really good position to deliver on our mission in retinal disease, which is to preserve and restore vision, both for the millions of people affected by common eye diseases, as well as for those with rare inherited retinal disorders. We are focusing on areas where patient need is greatest and where we believe Janssen can make the biggest impact. From rare inherited conditions to age-related macular degeneration and other conditions. Our current focus areas include rare inherited retinal diseases, including X-linked retinitis pigmentosa due to variants in the RPGR gene, as well as achromatopsia targeting forms due to variants in the CNGA3 and CNGB3 genes, and more prevalent conditions, sometimes called common eye diseases, including age-related macular degeneration, including a late form known as geographic atrophy, diabetic retinopathy, diabetic macular edema, and glaucoma. We have 5 important clinical programs underway at various stages of development, and a large proportion of our investigational assets are gene therapies. Our work here first began with our partnership with Mira GTX, who possess deep knowledge in the area of gene therapy and inherited retinal disorders, as well as relationships with hospitals and medical centers who work in these areas, along with the dedicated patient volunteers. We've had ongoing conversations with patients, caregivers, and advocacy organization leaders to ensure your voice is heard as we design our clinical trials and future resources. This is an exciting time to be at Janssen and with you, all of you today. I appreciate your time and attention and hope you found this to be helpful. If you'd like further information regarding our clinical trials and clinical studies, as Ben mentioned previously, please visit clinicaltrials.gov or reach out to your contact at Foundation Fighting Blindness, and we can work together to answer any questions you may have. We encourage you to speak directly with your healthcare provider for information specific to the right treatment for you and on genetic testing options. Thank you, and really appreciate the opportunity to meet all of you.
Thank you, Dr. Anglade. That was a nice overview of Janssen's focus in our space. We really appreciate some of the clinical work you're doing in X-linked RP and achromatopsia. And if you want to learn more about some of these trials, as Dr. Anglade said, you can go to clinicaltrials.gov. You can also visit the Foundation Fighting Blindness website, fightingblindness.org, to read some articles or listen to some articles on those. So with that, let's kick it over to Dr. Yang. Now, Dr. Yang, you're up. Sorry about getting things out of order. And while Dr. Yang is loading up his slides or sharing his screen, I'm going to give you a short bio. Dr. Yang is an assistant professor in ophthalmic genetics and immunology at the KCI Institute, which is at Oregon Health and Science University, where he specializes in patients with inherited retinal degenerations, autoimmune retinopathy, and gene therapy associated uveitis. He's a principal investigator and sub-investigator on numerous gene therapy and neuroprotection clinical trials for inherited retinal degenerations. And with funding from the FFB TRAP program, which I told you about, Dr. Yang continues to conduct translational research in his lab with the goal of bringing new medical treatments to the clinic for patients with inherited retinal degenerations. And I just wanted to comment, a few months back, I interviewed Dr. Yang for one of his projects, which I don't think you even cover in your talk. And when I realized how much clinical trial work you were involved in, I knew you were a natural for this webinar. So great to have you.
Great, thank you. Can you see my screen fine? Yeah?
Yes.
Okay, very good. Well, I want to thank Ben and Foundation Fighting Blindness for inviting me to participate in this event. It is my pleasure to talk to you about gene therapy clinical trials and inherited retinal degenerations, or IRDs. Advance, there we go. These are my disclosures. As Ben said, I am a consultant as well as investigator on many clinical trials. Today, I will provide a brief introduction and provide updates from 3 clinical trials that we have been part of at KCI Institute, namely the AGTC clinical trial for X-linked retinitis pigmentosa, or RP, due to mutations in RPGR, the AGTC clinical trial for Chromatopsia due to mutations in CNGB3 or CNGA3, and the ProQR clinical trial for RP or Usher type 2 due to mutations in exon 13 of the USH2A gene. Well, let's start with a brief primer on the retina. This is a schematic of the eye in cross-section. Light comes in the front of the eye and gets focused onto the retina. The retina is the layer of nerves that line the inside of the eye and the back and relays light signals to the brain, allowing us to see. The light-sensing nerves in the retina are called photoreceptors, which are composed of 2 types, cones and rods. In general, the rods are responsible for night vision and peripheral vision, whereas cones are responsible for daytime vision, color vision, and central vision. Cones are concentrated in the part of the retina called the macula, where the 20/20 vision comes from. As shown here, this is a Venn diagram showing many retinal genes that, when mutated, are known to be associated with inherited retinal degenerations. One of these genes, RPE65, is the target of the first FDA-approved gene therapy, Luxturna, which was a landmark event in December of 2017. Over the last decade, there has been an explosion in the development of gene therapy clinical trials for other genotypes and inherited retinal degenerations. Highlighted here are the genes being targeted by 1 or more gene therapy clinical trials. Indeed, some genes are the target of clinical trials by multiple sponsors, such as REP1, which is associated with Coredovemia, RPGR associated with X-linked RP, and CNGA3 and CNGB3 associated with the Chromatopsia. For today's talk, I will concentrate on RPGR, CNGA3, and CNGB3, as well as USH2A associated non-syndromic RP, or Usher type 2. These clinical trials also illustrate 2 different approaches to gene therapy, gene augmentation and antisense oligonucleotides. Gene augmentation is the delivery of a normal copy of the mutant gene to the retina, typically using an adenovirus vector or AAV. Once delivered, the photoreceptors can use the new gene to make normal protein, leading to rehabilitation of its function and stopping or slowing of degeneration. Antisense oligonucleotides use specially designed RNA fragments that reprogram the way cells translate information from a gene into a protein. In the case of USH2A, the RNA therapy instructs the cell to skip and exclude a small section of the genetic instructions that are prone to mutations, specifically exon 13. The result is a slightly smaller but functional protein, leading to improved photoreceptor function and stopping or slowing of degeneration. These two strategies are also delivered differently to the eye. Gene augmentation with AAV is typically delivered via a surgery and injection of the treatment under the retina, specifically under the macula, to target the central vision area. That also means that the peripheral vision area is not typically treated. This strategy is thought to be a one-time permanent treatment. Antisense oligonucleotides do not require AAV. Thus, it can be used to treat genes that are too large to fit in the AAV vector, such as the USH2A gene. The RNA fragments are injected into the cavity of the eye as a simple outpatient procedure. Theoretically, this method can treat both the central and peripheral vision areas. However, RNA fragments naturally degrade over time, so repeat injections would be needed. Again, there are multiple sponsors currently conducting clinical trials for these three conditions. But for the sake of time, I will only be concentrating on updates from the AGTC and ProQR clinical trials. First, let's talk briefly about achromatopsia. Achromatopsia is caused by a complete loss of cone photoreceptor function, which leads to severely reduced visual acuity accompanied by uncontrollable eye movements or nystagmus, severely reduced color vision, and severe aversion to bright light. As shown here in this figure, vision is without color and bleached out during the day, and even in the best of circumstances, the visual acuity is poor. There is no cure, and the only treatment is red tinted glasses, which helps to reduce sensitivity to light. Prevalence is 1 in 30,000, and the CNGB3 and CNGA3 are the most commonly associated genes. Now let's discuss the AGTC phase 1-2 clinical trials for CNGB3 and CNGA3 associated chromatopsia using AAV gene augmentation. So far, 25 CNGB3 and 19 CNGA3 patients have received a single subretinal injection of the gene therapy. Typically, for phase 1-2 clinical trials, both of these trials start by enrolling adults while slowly increasing the gene therapy dose across 6 groups. Additional cohorts for children occur later in the trial once minimal safety data has been shown. And as always, safety is the primary objective in these early trials. Secondary efficacy endpoints include visual acuity as measured on an eye chart, rod and cone function as measured by visual fields, light sensitivity tests, color vision tests, and quality of life questionnaires. The conclusion after 12 months of follow-up is that the gene therapy was safe and well tolerated. Gene therapy associated inflammation was well controlled in this study. The preliminary results demonstrate that there were reductions in sensitivity to light and improvements in cone photoreceptor function on visual fields. There was also subjective improvement on the questionnaires, with reports of decreased sensitivity to daylight and reduced need to wear tinted glasses indoors. Currently, the phase 1-2 trial for CNGB3 and CNGA3 are expanding into even younger pediatric patients, age 4 to 8 years of age. We theorize that younger patients may respond better to gene therapy because the visual parts of the brain may be better able to process the new information. Additional endpoints such as functional MRI and color brightness test have also been added in an attempt to better detect the benefits of gene therapy. All right, let's switch gears and now we'll talk about retinitis pigmentosa, or RP. RP is caused by a primary degeneration of the rod photoreceptors with subsequent cone degeneration. Early on, there is progressive night blindness and loss of peripheral vision, which causes large blind spots in the vision that is simulated in the figure here. In advanced disease, visual field constriction eventually leads to tunnel vision, loss of visual acuity, and legal blindness. There are no approved treatments except for RPE65, as I discussed earlier. Prevalence is 1 in 4,000. The USH2A gene is associated with 12 to 25% of autosomal recessive RP and the majority of Usher type 2. The RPGR gene is associated with the majority of X-linked RP and mainly affects males. Next, we will discuss the AGTC phase 1/2 clinical trial for RPGR-associated X-linked RP using AAV gene augmentation. So far, 29 male patients have received a single subretinal injection of the gene therapy with increasing dose across 5 groups. The primary outcome is safety, and the main secondary efficacy endpoints include visual acuity as measured by an eye chart, central visual sensitivity as measured with the visual field, the macular area. The conclusion after 12 months of follow-up is that the gene therapy was well tolerated and safe. Inflammation was mild and well controlled. Moreover, there was a sustained increase in the central visual sensitivity in 28 to 50% of treated eyes in the 3 highest dose cohorts. There was also supportive evidence of sustained increase in visual acuity of 1 or more lines on the eye chart in the treated eye. Patients also had subjective reports of improved vision and navigation under dim lighting, as well as seeing small things better and reading faster. Currently, there is a phase 1/2 expansion that is enrolling 12 more patients in a randomized and masked fashion to 2 different doses. There is also a phase 2/3 clinical trial being planned that will enroll 60 patients, randomized and masked to 2 doses versus no treatment. The enrollment criteria will be male RPGR X-linked RP patients age 8 to 50 and visual acuity of 20/30 to 2200. Next, let's talk about the ProQR phase 1/2 clinical trial for USH2A associated RP or Usher type 2 using antisense oligonucleotides. The phase 1/2 clinical trial has been completed and patients were randomized to either treatment or a sham procedure with no injection. In total, 14 patients received a single intravitreal injection of the treatment with increasing concentrations across 3 dose groups, and 6 patients received sham. Primary objective was safety, and secondary efficacy endpoints included visual acuity as measured with an eye chart, retinal or macular sensitivity as measured visual fields, and macular photoreceptor structure as measured with optical coherence tomography or OCT. The conclusion after 48 weeks of follow-up is that the RNA therapy was well tolerated and safe. There was no inflammation and only 1 case of pre-existing macular edema that had worsened. Patients with advanced disease, as determined by visual acuity of 20/40 or worse, showed stabilization of the visual acuity in the treated eye. Patients with better visual acuity were considered early to moderate disease, and these patients had an improvement in retinal sensitivity in the treated eye. In addition, there was also evidence of stabilization of macular structure and macular sensitivity in the treated eye. Currently, separate phase 2/3 clinical trials are being planned for patients with advanced versus early to moderate disease. Both trials will randomize patients to 2 different doses versus sham procedure. Enrollment criteria will continue to require patients to have 2 mutations in USH2A with at least 1 of them in exon 13 of the gene. So in summary, I have shown that many gene therapy clinical trials for inherited retinal degenerations are well underway. We discussed 3 programs in depth, wherein the preliminary data has been encouraging for safety and potential efficacy for gene augmentation for the CNGB3 or CNGA3 in a chromatopsia, RPGR and X-linked RP, as well as RNA therapy for USH2A in RP or Usher type 2. And if you want to get involved, please make sure you have an up-to-date clinical diagnosis, get genetic testing, and register with my retina tracker. I want to thank you for your attention. I'd also like to thank all the patients, my colleagues, staff, technicians, and coordinators who make this work possible. And I'll be happy to take questions at the end of the session.
Really nice talk, Dr. Yang. Thanks for giving an overview of those projects. And we're getting questions about some of your other trials that you've been involved with. So hopefully we can get to those. So we are moving on to our next presentation, our final presentation before the Q&A. And I'm happy to welcome Samantha Vieira to join us. And Ms. Vieira, you can begin sharing your slides. I'm going to give you a little background on Ms. Vieira. She's an MBA and Executive Director of Ophthalmology at Biogen. She's currently leading a global product development and commercial team to define and execute on an integrated, innovative, global commercial strategy for BIIB111, which is Biogen's lead clinical stage retinal gene therapy program. And I believe that's for choroideremia, if I remember correctly.
That's right.
Okay. Samantha joined Biogen from Nightstar Therapeutics. For those of you that have been following the choroideremia field, I'm sure you're familiar with Nightstar. They were a retinal gene therapy company. She was vice president, head of program management there, having joined early in 2015 as one of Nightstar's first employees. During her time at Nightstar, Samantha was involved in the portfolio and product development strategy and operations, the company's financing, and building out the team and successful acquisition by Biogen in 2019. And prior to Nightstar, Samantha spent just over 10 years in research and development, having held positions of increasing responsibility at Emergent Biosolutions within program management and Lonza Biologics in process development. So we can see your screen well. We can see your opening slide. So take it away.
Brilliant. Thank you very much. And good morning and good afternoon to everyone from various locations. I'm actually joining you from London. And thank you very much for the invitation to participate. As Ben mentioned, I'll try and put it a little bit simpler. So I've been working in drug development for just over kind of 10 years. But the last 6 of those have been within the inherited retinal dystrophy space for working on gene therapies. And I really love what I do, and I kind of love having the opportunity to participate in these types of events with participants that actually make what we do even more exciting. So today I'm going to give you a little bit of a brief background and overview of Biogen and our 2 clinical programs for inherited retinal dystrophies, which is choroideremia and XLRP, which we refer to as BIIB111 and BIIB112. So a little bit about Biogen. So it's actually a company that's over 40 years old. I'm not sure that many people realize that it was one of the first biotechs. And the company's really grown over the last 40 years. You know, we have over 9,000 employees globally. And what does that mean? That means we can run clinical studies and enroll participants from multiple locations. The company is really focused on a number of different therapeutic areas. And one of those growing areas is ophthalmology and gene therapy. You know, there's a lot of significant unmet need in a lot of these areas. You know, a lot of participants and patients don't have treatment options today, but there's a lot of opportunity. And we heard already about some of the progress that's being made in this field. And that's why I'm really kind of excited to be working in this space. So I'm going to show you a slide. So this is just a table showing Biogen's growing gene therapy pipeline. So you can see there, I mentioned the focus across different therapeutic areas, which includes ophthalmology, neuromuscular and muscle diseases, neurodegenerative diseases, and genetic and neurodevelopmental disorders. But today I'm going to focus on ophthalmology. And you can see at the top there, the slightly longer bars, which show that BIB111 for Coredovemia and BIB112 for XLRP were programs that were brought in when Biogen acquired Cena Therapeutics. And those are the two programs that we currently have in clinical trials. I will say that Biogen is also investing in manufacturing and other capabilities through collaborations with other companies, but also internally, so that we can also advance some of these other programs that you can see are still in preclinical testing. So let me move on to my next slide. So I'm going to start with Coredovemia. And Coredovemia is a really slowly progressing disease. You know, it initially presents in childhood with night blindness. Participants have trouble navigating in low light conditions. And then as people get slightly older, it progresses over time and patients start to lose their peripheral vision until it slowly encroaches on your central vision until ultimately you have severe vision loss and complete blindness. And I'm showing this because I wanted to raise the importance of natural history. And you'll see a lot of studies run a lot of companies, run natural history studies. And so in those studies, you know, there isn't an intervention, so there isn't a gene therapy or drug applied. But the intent is really to help us as companies understand, you know, the progression of the disease so that we're able to better design the clinical trials where we are introducing some of these drugs or gene therapies. The second piece that's really important, and it's been mentioned before, is around genetic testing. You know, in order to be enrolled in these studies for gene therapies for many of these inherited retinal dystrophies, getting the correct diagnosis of choroideremia in this case, and you know, ruling out some of the other retinal dystrophies is really important. At the moment, we're looking at roughly up to 60 to 80% of people with inherited retinal dystrophies that we can actually identify what the mutation is. And then, as I mentioned, you know, in addition to actually establishing what your correct diagnosis is, genetic testing can give you more information on the implications, but also importantly, allow people to participate in the appropriate clinical trial. So a little bit more about BIB111, which is what we call our program for our product for gene therapy. So BIB111 is a recombinant adeno-associated virus. Basically what that means is we use what's called an AAV2 vector, which is designed to deliver a functional version of the missing gene into the back of the eye. And over here, I've shown a little bit of the progress of BIB111. We started with phase 1/2 clinical studies. We then started a natural history study called NIGHT. So as I mentioned, really important to understand the progression of the disease to help us further design future clinical studies. We then also started a phase 2 clinical study called Gemini. And in that study, we're looking at treating both eyes to understand what the safety and the efficacy is treating 2 eyes. And then we had a phase 3 study called STAR, and I'll tell you a little bit more about that study. And then we also had a study called Solstice, which is a long-term follow-up study. As mentioned before, I think earlier, you know, we ran through the various stages of clinical development. And here I've also outlined what those stages are that we followed for BIB111, but also highlighting, you know, the natural history and then long-term follow-up. And the importance of that is that we can actually track what is happening after you've been treated with a potential therapy longer term. So BIB111 for choroideremia is in what we call phase 3. We've actually completed enrollment, and we couldn't have done that without the participation of many of you, of our patients and the caregivers that help them with participating in the clinical trial. So the STAR study, as we call it, was a phase 3 multi-center randomized 3-arm parallel control group study. And that's a little bit of a mouthful, but all that means is that it was a phase 3 study that we ran across multiple centers across the US and in Europe. We had 3 arms in the study, so a control group and 2 doses were tested in the study. And then what we're testing in the study is really looking at is BIM-111 efficacious and is it safe? And that's the endpoint. And in order for us to understand that, we have a particular endpoint which is called BCVA, so best corrected visual acuity. And so what we're doing is we're looking at 12 months after treatment, what's the proportion of patients or participants that have seen an improvement in their visual acuity. And so that's a little about the phase 3 STAR study. As I mentioned, it's fully enrolled, and we are expecting to have data that we'll share as soon as we can with all of you by the end of the first half of this year. Our second program that's in clinical testing is what we refer to as BIM-112, and that's for X-linked retinitis pigmentosa. And actually last week we announced data from a phase 2/3 study. And earlier in the talk, somebody spoke about endpoints. So we had a number of different endpoints and measures that we were looking at in our XLRP study. Unfortunately, we didn't meet the primary endpoint, and that was looking at the measure of retinal sensitivity. So it was a microperimetry measure in the study. So it didn't show statistical significance. However, there are secondary data that looked fairly promising. And so we're continuing to analyze the data and we'll be communicating what are the next steps for that program once our teams have had more time to assess the data and plan next steps. And so again, as soon as we have information that we can share, we will provide that to all of you. So just want to thank you once again for the time and the opportunity to give you a little bit of a snapshot of our programs at Biogen. Thank you.
Great, Samantha. That was a nice overview of your choroideremia gene therapy trial. I know we have a lot of people out there that are excited about the progress being made there. So thank you. So now I'd like to ask our other presenters, Dr. Yang and Dr. Anglade, to join us, as well as our Chief Scientific Officer, Dr. Gelfman, so we can begin the Q&A. One general comment that I'll make, we get literally hundreds of questions in the Q&A, and we appreciate those. The questions that we do not get to today, we will answer at a later date. It might take a little while, so please be patient. Like the science, answering the questions takes some time. But let's get things started. And I do have a favorite question of the afternoon here. And the reason I'm asking is because we've gotten a few questions along these lines, and the spectrum around this question is really heartening. So people are wondering what age range you need to be in to participate in a clinical trial. One person said, asked if somebody could participate if they were 97 years old. And another person asked if they could participate, or if someone who was 2 years old could participate. So I thought that was a pretty compelling age range. So does anybody want to take that question on?
Yeah, sure. I can take that question. It depends on the clinical trial, first of all. Some clinical trials have a lower age limit, but not a upper age limit. Some clinical trials have both an upper and a lower age limit. And typically, the lower age limit, as we mentioned earlier, is usually for the early phase trials where we want to do the initial dose escalation and the safety studies in adults. But usually, there are later cohorts where it opens up to pediatric patients. Now, the question of 2 years of age, all of these procedures, have risks and side effects to them. And that's not only the gene therapy, but also the actual procedure, the surgery to do the treatments. And so, there has to be a weighing of kind of the risks and benefits in enrolling children and how young to go. And so, that's considered, for each of these clinical trials. And, that's how they come up with that age limit. In the Chromatopsia clinical trials, as I mentioned, they've gone down to as young as 4 years old. And that's because they wanted to balance, the risks, right, of doing surgery and treatments in a young child, but also knowing that the younger you treat, probably the more likely you're going to see a biological signal or an effect. As far as the upper age limit, what a common upper age limit is, that I see is 50 years of age in the Stargardt clinical trials. And that's probably there because they don't want there to be any confusion between Stargardt patients and age-related macular degeneration patients. And that's because some of the neuroprotective clinical trials, they don't always require 2 mutations in APCA4. So again, it's just to make sure that they enroll the right kind of patients. But then in another clinical trial, such as the stem cell therapy clinical trial from Reneuron, you just have to have RP. Doesn't matter what your genetic mutation is, but you have to be greater than 18 years of age, and there's no upper limit.
That was a great comprehensive answer. Does anybody have any additional thoughts on age? Okay, so the next question is a very popular question, and I think has different aspects to it. And that's about genetic testing and also what to do if someone's gene hasn't been identified. And so let's start off with the second question I just posed about what do I do if my gene hasn't been identified? And I will assume for that question, somebody's been genetically tested with a pretty decent panel in recent history. So does anybody want to take that?
I can take that again.
Sure.
So first of all, I want to say that we are in a day and age here now that we are lucky to have genetic testing that's funded, and specifically right by FFB via the My Retina tracker. Not long ago, it was really an uphill battle to get patients genetically tested because it's not often paid for by insurance. So there's been great progress on that front. And so much so that there are now actually multiple panels, being offered not only by FFB, but also by sponsors from different labs. And it's also important to know that all the different labs, they have a somewhat different set of genes that they test. There are some differences. There's a lot in common among the different panels. So it's important, first of all, to talk with your specialty provider and genetic counselor to truly understand which of the panels was used for your genetic testing, and to understand what it means to be, have an inconclusive or negative result. And to make sure that, there are not other genes, that are possibly in question that is not on your panel, but maybe on another panel, right? So first and foremost, understand your results and talk to your specialist and genetic counselor. Now, if it's truly the case that your diagnosis does not have a genetic confirmation, right? What that means, it's just not yet known. We do not yet know all the genes that are associated with every single condition. Okay, so it's just not possible at this time to be 100%. In that case, it makes it hard, right, to be eligible for a gene therapy clinical trial, because by definition, you have to know what the genetic mutation is in order to be eligible for a genetic clinical trial. And in these cases, what I recommend is to continue following up with your specialist and, probably attempt to get retested in about 3 to 5 years once there's been enough improvement in our knowledge. These genetic testing panels, they keep growing from year to year, and our knowledge keeps getting better. Okay, I think that answers the question.
That was a nice answer. I don't know if anybody else wants to chime in and offer additional thoughts on.
Oh, I actually forgot one thing, Ben.
Okay.
As I mentioned, clinical trials like Regeneron, stem cell based treatments, don't usually require knowledge of genetic mutation, right? You just need to have a clinical diagnosis of, for example, retinitis pigmentosa. So, there are clinical trials that don't require knowledge of genetic mutation. So, so patients, who don't yet have that, could still qualify for those other clinical trials as long as they fulfill all the other criteria.
Yeah, and I might just add to that that the benefit of having the genetic testing and having your genetic milieu, if you will, deposited into the registry that we at FFB do, that's really important, because as more and more information is deposited into the database, as more mutations are identified, if your pool of your genetic material is in there, then and we periodically look to see, we essentially mine the database with new information we have around mutations that are constantly being discovered. We'd hate for anyone to miss out on that identification and the potential participation in a future clinical trial, just because at the time that you got genetic tested, your specific, the mutation was not known, but it could be later on. And to your point, Dr. Yang, as well, the information that goes into the registry is also all about the clinical manifestations that you're living with that can help also enroll in a clinical trial for some of these, what we were calling before, gene agnostic approaches. If it's known, for example, that you have cone malfunction or rod malfunction or some specifics, you not knowing your specific mutation could still qualify you for enrollment in a clinical trial. And, I think it's a great value that we offer through the foundation. I'd hate, none of us, we would all hate for anyone to miss out on participation in a clinical trial.
Yeah, great point, Dr. Gelfman, and nice answer, Dr. Yang. What I will add to this discussion is, if you're interested in pursuing our no-cost genetic testing program, there's information on it at fightingblindness.org. And it's a test that ultimately your doctor orders. And you can download documents that you can give to your doctor. They order it through Blueprint Genetics, our partner. Genetic counseling is part of that service, free genetic counseling. And then, as Dr. Gelfman said, if you can get your information into my retina tracker, you can get on the radar screen of researchers and clinicians doing clinical research. And I will add that my retina tracker is really independent from the genetic testing side of it. If you don't know your gene, if you got tested outside of our program, whatever, you can still be a part of my retina tracker. So while we talk about them together, they don't depend on one another. So again, look to our website for more information. So another general question that came across that I think is important to talk about. Someone asked, how does someone know if a clinical trial is safe? And I think that's, again, a really good question if somebody wants to address that.
I can start and then happy for the others to kind of add on. That is a really, really great question. And I'd say, you know, when I think it was Dr. Gelfman presented at the beginning, she spoke about preclinical testing. How do we know it's safe? In order for us to get approval to start a phase 1 or a clinical study, we have to show preclinical data. So that's data either in animal models or different cell types to show kind of, you know, early indications of safety. The other kind of key aspect that happens to get approval to start a clinical study is there's also ethics review boards that review the data, what we're trying to do, the outline of the study. And so it's quite I'd say a really rigorous process before we get approval. And then ultimately, you know, I think what was mentioned that the clinical trials are, you know, we're looking at efficacy and safety in participants. But happy if anybody else wants to add on to that.
And I guess the only thing I would add is that it's the paramount piece of every clinical trial, right? And there's, for every clinical trial that is even proposed, there is inclusion criteria and exclusion criteria. So inclusion criteria is everything that you kind of need to have before you enter the trial. The exclusion criteria is the opposite of things that if you've got it, you cannot enter the trial. And the reason for that is exactly what you were saying, is so that there's nothing that may be part of your, you know, if for example, you have some other situation, some other condition that in combination with the therapy that's being evaluated in the trial could cause anything harmful. You know, we want to mitigate that at the beginning. We want to reduce any possibility of any safety issues that could occur. And keep in mind too, that by the time a therapy gets to the point of being even evaluated in a human clinical trial, there's been very, very rigorous testing in animals, all types of species at all different doses, so that the doses that are given to humans in the human clinical trial, hopefully, and not just it's been verified under similar conditions to be safe. So that is incumbent upon us as a researcher, as a clinician, as a company, as an academician, to design the best study that will be safe for anyone enrolled in the clinical trial.
Excellent point. And that's, you know, safety is what the FDA is all about at the end of the day, or the EMA in Europe. So as long as a trial has some monitoring by a regulatory agency like the FDA or the EMA, you can at least feel confident that safety is being is a big factor. There are some quote unquote trials that are able to operate outside of the scope of the FDA, especially in the stem cell space. And patients are being charged for these alleged treatments, and those are things that need to be avoided because safety can be a big risk there. We got a couple of questions about a topic that we could spend a whole 2-hour webinar talking about, but I thought it would be good to cover the topic at least a little bit, and that's dry age-related macular degeneration. It's still an important unmet need. There's some good things happening in the wet AMD space. We do have FDA approvals for wet AMD, and Dr. Gelfman came out of that space, the wet AMD gene therapy space. But maybe we could spend 5 minutes talking about what's going on clinically for dry AMD.
Sure, I'm happy to start the discussion there. So, unlike wet AMD, you can actually have dry AMD and not know it. So there are different stages of the dry form that start with a very early, an early stage, an intermediate stage, and it's the late stage of dry AMD that causes blindness. And that's also known as geographic atrophy. So the geographic part of it, the cells that are truly atrophying, that are causing blindness at the late stage, it's a single layer of epithelial cells at the back of the eye, just beyond the retina, called the retinal pigmented epithelium. And when those cells atrophy, they cannot provide the support that the neighboring retina needs. And so then the retinas die, the photoreceptors, the rods and the cones begin to malfunction. And you can see from there, then we cannot capture the light appropriately coming in and then send a message to our brain to interpret the light coming in for proper vision. So there are a lot of companies in the space of looking for ways to modulate different parts of the pathology associated with dry AMD, particularly that late stage geographic atrophy. So in order to design a therapeutic to target that stage, we need to know what's behind that pathology, what's really going on at the cellular level. And so there's been a lot of important information that's come out of many different academic labs and companies that show that inflammation is part of it. There's immune response that's part of it. Their complement pathway is part of it. Oxidative stress is part of it. There are many different mechanisms that have been shown to go awry during that late stage. And there are many different companies that are pursuing treatments. Sometimes they're gene therapy based, sometimes they're small molecule based, sometimes they're protein therapeutic based to target some of those mechanisms, because probably the most important thing to realize about dry AMD, in contrast to a lot of the inherited retinal diseases that we've been discussing, is that we don't know the genetic cause of dry AMD. We don't know the causative gene where we can go in and restore a good copy of it, for example. So it's a much more difficult disease state to really work towards a therapy. There's a lot of work going on. That's the great news. There's so much that we're learning. There's a lot of companies that are in this space, but it's different in that it's considered to be multigenic. There are probably many genes involved, probably environmental factors that are involved. It's not as quote unquote simple as restoring a good copy of a specific gene.
Thank you. Does anybody want to add a little more, or are we good there? Okay. What I will suggest is people can go to clinicaltrials.gov. You can search under dry AMD or wet AMD. Advanced dry AMD is known as geographic atrophy. So if you have advanced dry AMD, you can search under that as well. And you can see some of the clinical trials underway. There are gene therapies and stem cell therapies moving forward for dry AMD and GA. So one of the proverbial questions that we get, but it keeps coming back, so we're going to address it again, is will participating in one clinical trial prohibit somebody from participating in a second clinical trial?
I can take that one. So I think it depends on the what that first clinical trial is. In general, stem cell-based treatments and gene therapy, gene augmentation, gene editing, those are considered treatments that are permanent. They cause a change, permanent change to the disease process, and hopefully there's benefit to that. And so if that is the first clinical trial, then often that means you cannot participate in a second clinical trial because the natural history of your disease has been altered, and it can confound the study of whatever that second clinical trial is. However, that's not to say that if a given treatment becomes FDA approved, right? It couldn't be, it could be still applied on a case-by-case manner to you in the, in your untreated eye, in the future. Let's see, was there a second part to that question?
Not really.
Okay.
I think, the bottom line is people, once you go into a clinical trial, it could prohibit you from participating in another trial. It just depends on, as you said, Dr. Yang, what that first treatment is. If it's something, a drug that can wash out in the first trial, then you probably have a better shot at participating in a second trial. Where a gene therapy trial, the gene is there forever, at least theoretically. So
right. And so that's that second point is important, is that the if the first trial is some sort of agent that has a half-life, right? And one of the ones I talked about, for example, the RNA treatments, we know that RNA will degrade and there's a half-life to it. Then there's a possibility that you can still participate in the second trial. And the same goes for other neuroprotective regimens that are injected or taken by mouth. Those we think have a half-life to it too. But obviously, that's all going to be up to the second clinical trial, right? It's whatever exclusion, inclusion criteria are in the second clinical trial that will govern, what can be done. And that is, kind of a little bit looking into the crystal ball. What's going to happen in the future, we don't know.
Right. Thank you. So I'd like to conclude with one more question. And this has been a question we've been getting a lot over the past year. And it's sort of I'll include two parts to the question. The first part is obviously our medical system, our biotech system did a wonderful job getting the COVID-19 vaccine through the pipeline and out to people. It's pretty remarkable how fast that moved. So the first question is, are we learning from that and taking that knowledge and applying it in our space, which is a very different space? So let me just ask that question to start, if anybody wants to tackle that.
So I think we're learning every day. I think we've learned from COVID, but I also think, as we presented a number of different studies going on by different companies. I think all of it is actually contributing to our knowledge and how we can do just improve on what we're doing. That's kind of my view on that, Ben. We're learning every day.
Exactly. I think it's what we're doing obviously is quite different from COVID, but it's actually I think quite more complicated. We aren't just looking for what the rate of infection is, right? In one, in the treatment group versus non-treatment group, we're dealing with very heterogeneous disease population, many different diseases, many different genotypes, which all require optimization of the endpoints. And so we are constantly trying to come up with the best endpoints that will tell us efficacy, right, as quickly as possible, right? That all kind of relates into the timing of how long it takes, how much follow-up does it take to determine whether a treatment, right, is efficacious. You have to have the right endpoints. And so there's a constant learning, that goes on where we're trying to develop those and optimize those. From the point of view of, FDA, I do have to say, if we look back at Luxturna, the Luxturna, FDA approval actually was quite quick. It was pretty fast. It was less than 1 year. Can it get faster? I don't know. I mean, that's already pretty quick. That was done under a, orphan disease kind of protocol. They knew there was an urgency to get this to market. But from all fronts, I'm sure everything can be improved, and we're all working hard to try to make things better.
Right. Thank you. So the second part of my question, and I'll preface it by saying I'm always impressed with how inquisitive and knowledgeable our donor and patient community can be. So I briefly referenced messenger RNA when I was talking about the ProCure therapy, because we're targeting therapies, or ProCure is targeting therapies, and Dr. Yang is working in this field toward messenger RNA. But in COVID-19 vaccines, messenger RNA are being used as a key mechanism of I guess pathway mechanism of action to get the vaccine to work. So they're two different things, but people are often asking about that. I don't know if folks on our panel want to talk about messenger RNA and compare and contrast what's happening in the retinal world versus the COVID vaccine world.
Yeah, I'm happy to jump on that. So, right, when you spoke, Ben, about messenger RNA, you were describing how it is made from a piece of DNA, and then how that messenger RNA is then goes on to make a protein, because it's really properly functioning proteins in all the cells in our body that keeps us all healthy. So when there's ever a mutation in the DNA and the RNA can't be made and the protein can't be made, there are issues. So when we think about, for example, these antisense oligonucleotides, this is a piece of a nucleic acid that's going to come in and essentially cover up that mutation in the mRNA, so the wrong protein is not made. The correct protein can still be made. So we're manipulating the system in that way, using a piece of nucleic acid at the level of the RNA, so that the incorrect protein will not be made and we can restore a properly functioning protein. So in contrast to mRNA vaccines, like the COVID vaccine, the protein in question is something called the spike protein. You may have read about it. It's in the news now. And the spike protein is actually, it lives on the surface of COVID-19, on that virus. And so what we're doing is we're actually, the virus itself, the vaccine itself is an mRNA that's telling our cells to make that's a piece of that spike protein. And our bodies haven't seen that before. So there's an immune response. And it's fairly active in some of us, when we got vaccinated. But what that does, it primes up. It's like armory. I think of it as, now we have armor to protect us when we get infected again later on. So it's manipulating at the level of the RNA, in one case, to prime our bodies to be prepared for an attack later on. And in the other case, like in the case of ProCure, we're actually using a piece of RNA to cover up a mutation so that our bodies can still make the correct protein.
That's a great description. So in the COVID vaccine, it's sort of telling your body that, hey, this COVID virus may be headed your way, and the body's putting up a defense system so that if someone is really exposed to COVID, their immune system is primed and ready to go. Does that sort of make sense? Okay. That was my attempt to put it another way. So anyway, we are nearing the end of our program here. So let's stop the questions. I really appreciate Dr. Yang, Dr. Vieira, and Dr. Gelfman for fielding that really diverse body of questions. You all did an outstanding job, and I appreciate your presentations as well. But now I would like to turn it over to our webinar closers from our national chapter organization. And those closers are Nathan Kluke and Bailey Bosselman. And they're doing a great job leading our newly revamped and expanding chapter program.
Thank you, Ben. As Ben mentioned, my name is Nathan Kluke, and I'm the Director of Chapter Engagement for the Foundation. And I want to invite you all to join us for our National Virtual Vision Walk, which is coming up on Saturday, June 12th. So grab your friends and family and join us to support the Foundation's mission. For more information or to sign up for the walk, you can visit fightingblindness.org/visionwalk.
Wonderful. And like Ben said, my name is Bailey Bostelman, and I am the Director of Chapter Engagement for the East. So our local chapters are the backbone of the Foundation's mission, and we focus efforts on education, resources, and revenue. Not only do our chapters provide the needed support for those in the community, but they also hold seminars and meetings which provide research information, low vision resources, and other industry topics, as well as provide outreach to retinal practices and eye care professionals. For information about joining one of your local chapters, visit us at fightingblindness.org/chapters. Or if you're interested in learning about the chapter leadership roles, email us at chapters@fightingblindness.org. Also, be sure to join us on July 17th for our nationwide virtual chapter launch event. This event is open to all chapter leaders and anyone interested in volunteering for their local chapter. We will talk about hosting local chapter meetings, community outreach, and also leave some time for networking with chapter leaders from across the country. Once again, thank you to all of our presenters today, and thank you for everyone for joining us. For questions that were not answered during the Q&A portion, we'll be following up separately at a later date. We will also be sending out the recording of this session, as well as have it available on our website and social media for future viewing of today's presentation. Have a great weekend, everyone.
So thank you, Nathan and Bailey. Thanks again to our presenters. And most of all, thanks to everyone out there for joining us and for supporting our urgent mission. Have a great rest of your day or evening.